have our single cell suspension on ice, and now we're ready to prepare the master mix. In the meantime, we have taken out our gel beads from -80 degrees C. The gel beads need to equilibrate to room temperature for at least 30 minutes. First, we have our RT reagent B. We equilibrate to room temperature, vortex, verify that there is no precipitate, briefly spin down, and leave at room temperature. Then we have our template switch oligo. Briefly spin down and resuspend in 80 microliters low TE. Vortex for 15 seconds at max speed, briefly spin down, and leave at room temperature for at least 30 minutes. Next, we have our reducing agent B. We equilibrate to room temperature, vortex, verify that there is no precipitate, briefly spin down, and leave at room temperature. Finally, we have our RT enzyme C, which is kept on ice. Briefly spin down before adding to the master mix. We're now ready to prepare the master mix on ice. We will add the reagents in the order shown in the user guide. First, we add RT reagent B, then template switch oligo, then reducing agent B, and finally RT enzyme C. We pipette mix 15 times and spin down briefly. Then we will aliquot master mix into each tube of a PCR 8-tube strip on ice. We've now prepared our master mix and are ready to assemble the chip.